If one day we held the pen of God, what would we write? This question, echoing through the corridors of science and philosophy, invites us to imagine a future where the boundaries between creator and creation blur. Picture, if you will, the intricate double helix of DNA, swirling in a dance of cosmic complexity, a code, ancient and profound, that has shaped life on Earth for billions of years. Now, imagine laboratories filled with the quiet hum of machines, vials of possibility, and the sharp focus of scientists gazing through microscopes, poised to edit the very essence of life. The music of discovery plays softly in the background, minimal and haunting, as a new chapter in human history begins to unfold. At the heart of this revolution is CRISPR, a word that, in just a few years, has leaped from scientific journals to the headlines of the world. But what is CRISPR really? In its simplest, most striking definition, CRISPR is a tool that allows us to edit genes with unprecedented precision. It is a molecular scalpel capable of cutting and rewriting the code of life itself. With CRISPR, humanity has gained the ability not only to read the story written in our DNA, but to edit it, to erase, correct, or even insert new lines into the narrative of our biology. But how does CRISPR actually work? The story begins deep within the microbial world, where bacteria, in their eternal battle against viruses, developed a clever defense mechanism. They learned to store snippets of viral DNA in their own genomes, creating a molecular memory of past invaders. When the same virus attacked again, the bacteria would use these stored sequences, along with a protein called Cas9, to seek out and destroy the viral DNA. Scientists, led by Emmanuel Charpentier and Jennifer Doudna, saw the elegance of this system and wondered, could we harness this natural machinery to edit the DNA of any organism, including humans? In 2012, Charpentier and Doudna published a groundbreaking paper outlining how CRISPR-Cas9 could be programmed to target specific DNA sequences. By designing a short piece of RNA to match the target gene, scientists could guide the Cas9 protein to the precise location in the genome where it would make a cut. The cell's natural repair mechanisms would then spring into action, allowing researchers to either disable a gene, correct a mutation, or even insert new genetic material. This discovery, which earned them the Nobel Prize in Chemistry in 2020, has opened a new era in genetics, one where editing the code of life is as simple, in principle, as editing text on a computer screen. The implications are as vast as they are profound. In medicine, CRISPR offers the promise of curing genetic diseases that once seemed untouchable. Imagine a child born with sickle cell anemia, condemned to a life of pain and hospital visits. With CRISPR, scientists have already demonstrated the ability to correct the faulty gene responsible for the disease, offering the hope of a permanent cure. Cystic fibrosis, muscular dystrophy, and hundreds of other inherited disorders may soon be treatable at their genetic roots, rather than merely managing their symptoms. The revolution extends beyond the human body. In agriculture, CRISPR is being used to create crops that are more resistant to pests, drought, and disease. The vision of rice that can withstand floods, or corn that thrives despite climate change, is no longer science fiction. These advances promise to feed a growing global population while reducing the environmental impact of farming. Even during the COVID-19 pandemic, the principles of gene editing and mRNA technology enabled the rapid development of vaccines, demonstrating how genetic tools can transform the way we respond to global health challenges. Yet, as with all revolutions, the genetic age brings with it new dilemmas and dangers. The power to edit life is, in many ways, the power to play God. Should we use this technology only to cure disease, or should we strive to enhance ourselves and our children? The concept of designer babies, where parents might choose the traits of their offspring, from eye color to intelligence, has leaped from dystopian fiction into the realm of possibility. In 2018, the world was shocked when a Chinese scientist, He Jiankui, announced the birth of twin girls whose genomes had been edited to confer resistance to HIV. The announcement was met with a storm of controversy, raising questions about safety, consent, and the very nature of what it means to be human. The specter of genetic elitism hovers over these debates. If only the wealthy can afford to enhance their children, will we create a world where social divides are written into our DNA? Will the playgrounds of the future be divided, not by class or ethnicity, but by genetic destiny? Some warn of a future where the rich design children for intelligence, athleticism, or beauty, while the poor are left behind, unable to compete in a world that values genetic perfection. Religious and philosophical voices, too, have joined the conversation. For millennia, the creation of life was seen as the province of the divine, a mystery to be revered, not manipulated. Now, as we learn to rewrite the code of creation, some ask, are we usurping a role that is not ours to claim? Or is the urge to heal and perfect ourselves an expression of the very creativity that defines us as human? In the words of one theologian, if we can rewrite our DNA, are we merely fulfilling our destiny as co-creators, or are we crossing a line that should never be crossed? As we gaze into the future, the questions only grow more daunting. Is genetic immortality within reach? Some scientists imagine a world where aging is not an inevitable decline, but a disease to be cured. A future where the genetic pathways that govern cell death and renewal can be fine-tuned, allowing us to extend life far beyond its natural limits. The philosophy of transhumanism, which envisions the merging of biology and technology, finds new fuel in the promise of gene editing. Could we one day design humans who are smarter, stronger, and longer-lived than any who have come before? The race is already on. Countries around the world, from the United States to China and Europe, are investing billions in genetic research. Some see a new kind of arms race emerging, not for nuclear weapons, but for genetic superiority. The nation that masters gene editing may hold the key to economic and military dominance in the decades to come. The specter of genetic warfare, where the genomes of populations could be targeted or manipulated, is no longer the stuff of science fiction. And so we stand at a crossroads, between hope and hubris, between healing and harm. The laboratories of today may give birth to the dystopias or utopias of tomorrow. Will we use CRISPR to eradicate disease, feed the hungry, and lift the suffering from their pain? Or will we stumble into a world where humanity itself is divided, reshaped, and perhaps even diminished by our own ambition? If we can rewrite DNA, can we also rewrite ourselves? This is the ultimate question, one that echoes through the halls of science, ethics, and faith. Will we use our new powers to heal, to help, to build a better world? Or will we use them to control, to dominate, to create a future where the essence of what it means to be human is forever altered? The answers are not easy. For every promise of healing, there is a peril of misuse. For every hope of progress, a warning of unintended consequences. The pen is in our hands now, poised above the book of life. And so, the question comes back to us, with all its weight and wonder. If we could write the story of humanity anew, what would we choose? Would we edit our flaws, erase our sufferings, enhance our gifts? Or would we find, in our imperfection, the very thing that makes us human? As the music fades and the images of DNA and laboratories dissolve into the background, we are left with these questions, unanswered, perhaps unanswerable, but utterly essential. Will we use gene editing to heal or to control? Should science race forward, or should it pause and reflect on the moral boundaries of its power? The future is unwritten, the code is ours to shape. The story of CRISPR is only beginning, and its ending, if there will ever be one, depends on the choices we make, together as a species on the brink of a new era. Subscribe for more videos.